Joseph Bump. I'm faculty in the department, Director of Grad Studies as well. Welcome to this Conservation Science Seminar, where we're welcoming Dr. Jennifer Smith, Jen Smith. Sincere thanks for making the effort to come here in person. It's great. And welcome to all of you who are tuning in online. Briefly, Dr. Smith's a behavioral ecologist with a lot of accolades in that field. Earned excellence in teaching and research, impressive record, particularly of work to diversify students and access to the field. I want to point out a couple of highlights. She's earned awards from the Animal Behavior Society with access to the field, and particularly strong scholarship, I would say, in the number of perspectives and techniques that she brings. She has extensive experience with large carnivores, especially spotted hyenas, ground squirrels, marmots, prairie voles. And lucky for us, something we should all know is that Dr. Smith recently relocated to the University of Wisconsin-Eau Claire, and proximity enables collaboration. So something that our labs, the Bump Lab and Smith Lab, are pursuing, and I would encourage any of you to explore the same. Today's seminar is on the comparative social evolution of mammals, and it'll cover a number of fundamental areas, from conflict to coalitions, from natural to human-caused impacts on foraging. I'm looking forward to it. Please check out Dr. Smith's biosketch, and a link is there provided to her impressively curated webpage as well. And without further introduction, then, welcome, Dr. Smith.
Thank you. Great, thank you for we're getting our hybrid meeting going, and we are successful. Thank you for the introduction. So great to be here. I particularly wanted to join in person because I'm about an hour-and-a-half drive away, and I'm really excited to foster some collaborations with you all. And I have more data than I know what to do with. So you'll see I use large data sets, both long-term studies on California ground squirrels currently, and comparative studies across sometimes all species of mammals. Today I'll be talking about comparative social evolution, and I want to give you a little bit of a background of where I've been and where I'm going for future years. Computer's being a little slow. It did go forward. I'm going to pop out. And back in. Okay. It was in a cold car. Who knows? Great. So the types of questions that I ask are about collective behavior, group level behavior, and I'm interested in how individual variation influences this large scale type of behavior. And these slides, okay. One of the questions that I've spent a lot of time on is thinking about whether individuals across time are consistent in their behavior. And I'm generally interested in social interactions and how animals interact over time with members of their own social group. These are little 30-second slides that are, there we go. So we can follow groups of monkeys. For example, these are some capuchins, and these same individuals are negotiating lots of different things in their world. So they might be eating together, they might be forming a coalition. So to what extent do some animals, are they consistently social or less social across these different types of contexts? And I'm also interested in understanding how, most recently, sex differences may influence collective behavior. So are males or females more likely to influence the start of collective behavior? It seems that when I exit my meeting controls, it gets stuck. There we go. Where I've been in the past is to think about lots of different social decisions and ways that animals interact with their world. So We've got some folks with Michigan State background, so that's excellent. My dissertation work was on spotted hyenas, and I spent a lot of time thinking about in these female-dominated societies how two or more hyenas join forces to interact with one another, and to what extent they do this to support members of their genetic family groups. And they do this based on both maternal and paternal relationships. They're also incredibly fluid in terms of how they interact with each other over time. They're in these fission-fusion societies. So like humans, you came in here, you'll leave after. They're in these really fluid groups, and they can either spend time with each other with conspecifics or alone. And the degree to which they do this varies based on how much food is available. So getting a little bit covered up, but right on the bottom axis here, it shows different months of the year. And in the Masai Mara, where we do the work, there is a superabundance of prey in these middle months of the year, represented by these black bars. And what we see is that the social cohesion of these animals is reflected by how much food is around. So we first documented that general pattern, and then we go and looked at this in a little more detail. We started thinking about this in a social network context. And each of these little circles or dots represents an individual hyena. And the hyenas interact preferably with members of their kin groups. And they do this oftentimes when prey is of low abundance, but these social networks are very dynamic, and they actually vary across time, and they shift in how cohesive they are based on how much food is immediately available. And if I advance the slide, what you would be seeing is that another network pops up. It's much more cohesive. And there we go. Superabundance of prey coming in, migratory prey, zebra, wildebeest, etc. We have a much more cohesive social network. And then this is dynamic and responds again when those prey leave. The strength of these social connections are less strong, still preferring kin over non-kin, but these dynamics are quite fluid over time. And so we started thinking about this. We wrote this review paper at the time, which was pretty new, of thinking about animals in social networks and how those social network connections are dynamic and change with things like prey and other aspects of the environment, which are influencing really quite dynamic types of social relationships. I've also in the past thought a lot about comparative social evolution, primarily with carnivores. And a lot of these phylogenies that I'll show you will be difficult to see all of the different species, but each one of these rows reflects a particular species. And in this phylogeny, it's a set of carnivores. In this work, we're looking at different types of cooperative behavior. And the cooperation that we looked at are whether there's cooperative hunting, whether there's cooperative breeding, and other kinds of cooperation. What we found was that increases in brain sizes, reduction in sexual dimorphism, so males and females of similar sizes, and a diet based on meat, all of those things gave rise to more cooperation in these carnivores. Today, I'm going to give you a similar kind of more recent journey of things that I've been thinking about. And the first focus will be on social decisions that individuals within a long-term study on California ground squirrels make, and how those social decisions have fitness consequences for individuals. So these are larger than life ground squirrels if you're here in person. And what you'll notice is this bird looks like a McDonald's M, if you will. We paint marks on their backs, and we also give them an ear tag. And we do this because understanding who's who is incredibly important for the work that we do. We observe them with binoculars. They're out in the Bay Area. They're really easy to see. in these open areas, and this gives rise to lots of rich data sets. The second part of the talk will focus on these comparative questions, thinking across mammals. What are the evolutionary origins and what's explaining the diversity with respect to collective behaviors across lots of different types of mammals? And hopefully with the goal, it's aspirational, of thinking about what the broad general patterns are across mammals in general, and maybe even making some connections to humans, as we are also mammals and social. So I'll first take you to the Bay Area. So I was on the faculty at Mills College for the last 10 years and have now moved here. And I'll continue to bring students from here, meaning Wisconsin, continue to bring students out every summer, which is actually when we do the work. The squirrels are pretty, spending a lot of time below ground. They're not as active during the winter months, so it's very convenient for teaching. And what we do is we go out every year and we put these traps out on the ground, and we live trap squirrels at 2 different locations. This is in the San Francisco Bay Area, and looks like this after the rain, but it's usually generally pretty dry. No humidity. So that'll be a fun little escape too. Members of my team are primarily undergraduate. We have a very inclusive team. We have lots of folks that are involved in different types of projects. It's very important for me to understand where students coming from and put them on a project that will inspire them and help them to get to the next level. And we have a lot of fun. So Team Squirrel, and we foster this inclusive environment. We believe that science should not be just in a vacuum. We actually go out into the world and share natural history with folks out there, and try to make a welcome, inclusive environment. And fundamentally, we also believe that squirrels are cool. And these are ground squirrels that we're focusing on. So not the tree squirrels that you might see around here. So why are squirrels cool? Well, I've already told you they're really easy to work with. They are predictable. They're in a spot. The ground squirrels, they have a home. We can show up and we'll find them right away. They're also semi-fossorial. So what that means is they split their time between life above ground and life below ground. They're also facultatively social. So they vary in how social they are. So they can spend time alone, or they can be in larger social groups. And individuals vary in how they do that. Now, you're probably captivated by what's going on here in the upper left-hand corner. And these are 2 California ground squirrels. We've got Volcano and Pi. You can see their marks. They are forming a coalition against a rattlesnake. And this is something that they do quite well. And rattlesnake and other predation by coyotes and birds of prey has really favored the evolution of sociality in these animals. And they've really co-evolved with all of these natural predators. So they're a major prey animal for lots of native species in California. And I'm interested in some of their anti-predator behaviors. We study them at 2 different field sites, and they're incredibly close to each other. You'll see on the right here is a, actually, I guess I could point here so everyone could see. So on the right here, we've got a parking lot, and that's very convenient for us, so we can just drive right in. Our main field site here is a relatively disturbed site, and there's a rest area, some picnic tables, and there's actually a gradient of how many humans and dogs come into that area. Right down the walking path is a relatively pristine site that has very few dogs and humans that come through. And so this actually sets up a really nice opportunity to compare the ecology of the 2 different populations of animals. We have very little movement between the 2, even though it's very easy for us as humans to monitor both groups. So one of the things that I'm interested in is stress physiology. And we have looked at the differences after controlling for the sex of the individuals. So they're males and females that vary in fecal glucocorticoid metabolites. So we extract stress hormones essentially from the poop of these squirrels. They go into the trap, Every time the trap closes, they give us a nice fecal sample, and we're able to get all sorts of interesting biology from that known individual. And one of the things that we found is that in the low disturbance site, shown here on the left and here in blue, that there is on average a lower baseline stress level in these animals. And that's true for both males and females compared to animals living at the more disturbed site. I'm going to be bold and try to hide my pointer and see if I can still advance. We'll see. No, I can't. Okay, never mind. So what we've done more recently is that was part of our validation of the work. And we've done more recently, Zainab has taken the lead on this project. We've looked across our more disturbed site to see if there's really fine spatial scale variation in how stressed animals are. And in fact, we found that there is. So this is only in one site. They're all experiencing humans and dogs to some extent, but the ones that are experiencing them more often have much higher stress levels than do other animals. So we've got, this is our measure of stress, and once we control for a number of covariates, this is 1 year of data. We have a decade of data that we're looking at this in. You can see identity, who the individual is, matters so much that when we look at it, it almost looks like a shotgun blast of points. When we control for which squirrel we were measuring, where they were living, we see that their interactions with humans and dogs is incredibly important in influencing their stress physiology. And this is over just oftentimes several feet differences in where squirrels are living. So their worlds are happening on a much smaller scale than ours, which coming off of hyenas took me a little while to adjust to what squirrels are experiencing. And this matters. So ground squirrels that are stressed are less likely to be able to cope with rattlesnake venom. So we know that adult squirrels are able to mount this venom inhibition response in their blood. They can break down the toxins from rattlesnakes. Juveniles cannot. And so these adults will form coalitions on behalf of their offspring to intervene, and they can usually cope with rattlesnake venom. But what we found related to this is that how stressed, so low stress compared to higher stress levels, and this is on a log scale, but what we see is a negative relationship. So what that means is that squirrels that are lower stress, they have a greater ability to break down the rattlesnake venom, and that has powerful fitness consequences for the individuals. So there are possible links here between the way humans are influencing the stress physiology and then how that's in turn influencing the physiological responses to one of their major natural predators. We've also taken so that's sort of a physiological response, looking at how squirrels are responding to humans. We're also looking at behavioral responses, and this work is done by Chelsea Jimenez-Ortiz, who was my undergrad and is now in her final years of her PhD at UC Davis. And what she's been doing is she's been walking at squirrels. And so there's a really standard behavioral assay in behavioral ecology. It's called a flight initiation distance. So I could walk towards something. If I get close to it, it will fly away. And some individuals fly away. It's determined by the name came from work on birds. So they're flying away. The squirrels instead will walk away. So we walk towards. The squirrel is here and it will go away. And so under this, we will be able to drop flags as we approach individuals. And we ask, how close can we get to them? Do individuals vary in how close we can get? Are some more shy? Are some more bold? How do they respond to humans? And so this is something that people have done a lot. But what she wanted to know was, okay, that's just one part of the story. What are the squirrels doing after they fly away? There's other aspects of the anti-predator response, and are those other aspects correlated to that initial decision? So in other words, you can have a squirrel that's here and they have decisions to make. So after they fly or walk away, They may stop and look before returning to other tasks, such as foraging. They may go into a shelter, and then they have to decide when it's safe to come back out, emerge from that shelter, and then return to other tasks. So for a squirrel, staying alive is important from predators, but also foraging is important for survival and reproduction. What she wanted to know is, are all of these different aspects of the anti-predator response, are they correlated with each other, such that an individual has correlated behaviors across each of them? And in fact, she found that they do. So if we look at the flight initiation distance for an individual squirrel, identity is incredibly important. And who an individual squirrel is matters a lot in terms of where they fall on this flight initiation distance continuum. Some squirrels we can get really, really close to. Other squirrels, they fly away or run away right away from a far distance. That's correlated with these other aspects of their behavior, including the time to emerge from a burrow. If they let me get very close, they'll go into a burrow and they'll emerge right away. And that's different from the individuals that are more fearful of humans approaching them. Similarly, the stop-look distance is tightly correlated with that flight initiation distance. And so this is really interesting to look at all of these different behaviors, the way they're correlated, and how each individual is consistent within them, but also across each of these phases. We've also gone back to those social networks and looked at things like ways that different threats or individual humans coming in or dogs or coyotes are changing the structure of the social network. And here, this is work with Chinny Gall. She's at the University of Exeter. She's one of a postdoc that's working with me. We can do so many things in this virtual world, so collaborations are incredibly fruitful and positive through pandemics and everything else through computers. And we've been looking at these really short networks, 10-minute networks, 10 minutes before, 10 minutes after a predator goes through the study site. So the dots here reflect individual squirrels. You'll see they're not very connected, but it's only a 10-minute time slice. And we have thousands of these. So we're able to simulate them. We have adults and juveniles. And we have before a disturbance and then after a disturbance, when a coyote, for example, shows up, there's rewiring in the social network. And on average, the same individuals that were interacting before a disturbance are more likely to interact after a disturbance when a coyote shows up. So in other words, coyotes are promoting social cohesion in our study site. However, we see a very different effect, and we thought it might be fairly similar. Coyotes and dogs, they're both canids, but when dogs and or humans are coming through, that tends to disrupt the social network. So the squirrels are less social than they were in the moments before the dogs or humans came through. We also found that both adults and juveniles were responding to the predator type, but adults were much more consistent in how they responded to each of these threats across time. And again, tie this back to fitness consequences as really important impacts on the lives of these squirrels. The degree to which they're able to aggregate and be social, so either foraging on their own or foraging with at least one other conspecific, influences how vigilant they are over time. So again, this is work by Chelsea. We can see that as group sizes increase, as squirrels are more social and are with more conspecifics, they spend less time looking. So vigilance is look, 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 and how often they're doing that. And they directly trade off between that looking rate and how much time they're feeding. How much time they're feeding influences how many offspring they can raise. And so if humans are disrupting social behavior, social networks, that can actually influence feeding rate as well. We're doing other things from fecal samples, and this is looking at microbiome diversity. So we just sent off about 300 samples to get analyzed to add to what I'll show you a little bit of pilot data. This is with Erin Person, who is a PhD student that's also getting close to finishing up. So several doctoral students are finishing up, and we need to have more collaborations if you're interested on Team Squirrel. Erin's work is looking at social behavior and microbiome and the connection between the two of those things. And we're at this point in the validation phase of the microbiome work. So we're finding lots of diversity of the genera that are in the poop. Importantly, what I wanted to make the connection for is that social dynamics are important for links to microbiome diversity. So if you look here, we can see that squirrels that have more social partners tend to have more rich microbiomes. And this is from a subset of squirrels, and we're doing this for more and more. So to think about humans coming through and disruptions that they might cause to sociality, that may also influence not only stress physiology, but the microbiome that's in there as well. And this is all non-lethal. They just every time they defecate, we can put that on ice, and then we can do the genomics to be able to look and see what's going on inside. Cool. So I'm going to switch gears a little bit. This is still focusing on ground squirrels. I want you to think about the different ways that we as humans interact, and maybe think about in sort of COVID lockdown time and how you're kind of sheltering in place in one office or room of your house and things like that, and how maybe we interact when they're in relatively confined spaces. Birds and other primates also do these things where they interact in these relatively confined spaces. The same individuals are doing it there. Hamadryas will go up to these sleeping sites and they'll escape from predators in these small spaces, even though they're in these big, vast cliffs, spending time together. And the question that I wanted to get at here from the squirrel work is, are these same individuals, as they move to other spaces, are the same ones that are relatively social in a relatively closed context? When they go out to the grocery store, they go out into the world, are they also the same, more social in that context? And we can ask these questions in lots of different species, but I was interested to do so in California ground squirrels because they have these two major ecological contexts that they have to deal with all of the time. So if you're a human walking around in the Bay Area, you probably see this above ground type of thing where you've got predators that we've been talking about, humans and all of that happening. But there's this whole other world that is below ground. And in fact, these squirrels are ecosystem engineers. They construct their architecture of their homes below ground, and they inherit these homes from one generation to another. And they're pretty complex, but we don't know, or we didn't really know before this study, what's going on down there. So taking a break for Watermelon Wednesday and work with Andy C. at UC Davis on this project. And in this project, we wanted to know is, what's going on on a social realm below ground, consistent with what they're doing above ground. So relatively confined space compared to one that's relatively open and free. So specifically, are the social traits consistent? So is an individual consistent within a particular context and how social they are within their social network? And what they're doing in one context, does that predict what they're doing in a second context with respect to how social they are? So below ground compared to above ground. So we've been doing, since the beginning of the project, we've been looking at these really conspicuous, friendly, affiliative interactions. And some of the networks I've already been showing you are actually based on these types of play slaps and play pounces and nose to nose greeting and nose to cheek greetings. And they do all of these really interesting types of behaviors that are really conspicuous. But we didn't really know what's happening underground. And so what we do is we do our typical thing where we go out and we live trap a ground squirrel. And you can see here's some of the fecal samples that I've been talking about. We just collect them in a trap. Here's Valesca Munoz with a squirrel that is marked on their back. And you can see some of the marks that we put on their back. They're incredibly docile. They're not anesthetized or anything. They just go there and they hang out and they're social. They're not like the tree squirrels. They're very, very docile. when they're handled, and they're in a dark bag that keeps them calm. We added for this project a PIT tag, passive integrated transponder, or like a microchip that you'd put on a dog to track them. And we also came up with this new radio frequency identification system for the squirrel project. So we have these antenna loops that sit in the entrance of the burrow. So every time the squirrel goes in or out of it, it scans them like a box of cereal at the grocery store, but instead it's a squirrel going in or going out. So we get directionality, and we get who it is, when it is, and who they're doing this with. And from these data, we first built up our traditional above ground networks. And so Minnie's here watching squirrels, and we have these different networks. We have different ones for different years. If we track the social behavior for an individual node within the network, you can see it's actually fairly consistent across time. We wanted to know, are they consistently social within a context across time? We also built up these networks based on their below ground networks. So here you can see the data loggers and the antenna loops that are situated here. It's recording all of this 24 hours a day, all year long. And we build up social networks based on that. And they're also consistent. In fact, their below ground social networks are more consistent than the above ground ones, but they're both statistically consistent over time. Yes. So the directionality is temporal. So the squirrels go to bed, they go into a hole, and then they emerge in the morning. You could do it a little bit more complicated, put two, one on top of another, and try to get directionality that way. But we've actually found that we're able to get it temporally by looking at their first emergence and then when they're going down. These data right here from the networks don't depend on that, but there are lots of questions that we could ask based on that, for sure. So the next question that we wanted to know is, okay, they're doing these networks below ground and we get consistent below ground structure, but to what extent does that actually predict what's happening above ground? Are these networks correlated? Are some individuals consistently social in both of these contexts? And in fact, we found that they are. And we ran this for multiple networks, multiple summers. And here's just a couple of examples of them, and you can see the structure. What's going on below ground is predicting the network structure above ground. So this suggests that some individuals are relatively less social and some are much more social. And this has really important implications for lots of different things, some of which I've already shown you. But I also wanted to show you a little bit of data that we've looked at more recently, which are the parasites, and we're starting to get into endoparasites. I was just telling Joe I'd hate to throw anything away. I have all these microbiome samples, like, oh, okay, we picked those out, we're good. But now I'm doing a new collaboration with endoparasites, so I'm so glad I didn't throw away the extra samples. That's one of the magical parts of these long-term studies is we often collect data for one purpose, and then you have all this information, you can go back and ask these questions you sometimes didn't even know were of interest or hadn't imagined in the first place. But one of the points that I wanted to make with this slide, I digress, is fleas. So we've been collecting fleas, we have thousands of trapping events, and every time we trap a squirrel the first time in that week, we comb them. So they're in this handling bag. We comb them 5 times, just a little flea comb that you might use on a cat or a dog. Put it into this bin. It's sprayed down with ethanol so they don't go anywhere. And we can count how many fleas an individual squirrel has. And some squirrels have a lot more fleas. So each one of these numbers here reflects an individual squirrel that we've trapped at least 5 times. And you can see the variation for, let's say, squirrel number 1 has some variation in how many fleas it has, but it consistently has more fleas than other squirrels in the population. And this is important because these fleas could potentially carry the bacterium that leads to bubonic plague. Now, our squirrels are healthy, but from a social network perspective and a disease dynamic and looking at zoonotics, it's really interesting to actually think about modeling how social network structure that I've been showing you might influence either the below ground or the above ground networks might influence the transmission of these fleas across time. So that's where we're going next. We know it's consistent. We control for a whole bunch of other things, highly repeatable. How can we explain it with social structure? Going back out big picture, I've been thinking about, all of these data and these different technologies that we're now able to use to understand the worlds of animals. I know you had, I think, shark behaviors come up in some of your more recent talks in this seminar series. A lot of these animals live in areas that are often difficult to monitor, but because of technology, we're able to put devices on animals or around in their areas and build up these really large data sets. So I'm super excited to think about ways that this will push the field forward with the intersection of, you're not going to ever get rid of, in my opinion, the muddy boot biologist, but to be able to push that forward with radio collars and all kinds of other technologies, I think is really, really exciting. And that intersection, I'd love to chat with you more about. What we're doing, so other work that Erin, the microbiome person, is doing is we're looking at how these social networks might influence food discovery in the area. So I've got a social network that is here on the right, and you can imagine that perhaps where an individual falls in that network might influence who they're watching to learn from. So what she does is she puts out these little plates. They have sand in them. She hides food in one of them. And these are also those antennas, they're a larger version, and they're scanning the squirrels as they walk across. And we can record whether they show up. And this is just the same one here. So you can see it more closely, whether this squirrel comes onto the plate, it records it. This one it's a baited plate, it has food on it. And to what extent, and this is an open question still, to what extent do the social network connections influence who's going to come next? So we would expect that animals that are socially connected might have more overlap in finding these food in this food discovery model. So that's one of the projects we're working on. We're also putting these collars on the squirrels. Squirrel collars are very, very tiny. They're a little bit frustrating. They don't last for very long, but they are offering some insights into the hidden spaces below ground. So the reason we started doing this is we're interested in training the collar, getting information that we can about the rate that the animal's walking. So we have accelerometers on here, a gyroscope if they're spinning, and all of these kinds of things that are happening when we see them above ground. We train the neural network then to try to uncover what's happening when they're out of sight, those same individuals below ground. And maybe we can start to map the architecture below ground, but that's maybe LiDAR is actually a better way to go forward. And this is some work with Amanda Robin, a PhD student at UCLA. And then this summer, one of our main focus, so each summer we try to have a main focus, and we're doing all of the long-term data collection, but we also have, experiments that we're running and so forth. And Sonia Wild has just come over from Switzerland. She won herself a 2-year postdoctoral fellowship to do work in my lab. And what she's doing is building these little puzzle boxes. And so here's a ground squirrel, and then this is a touch screen, and she's getting ready. At this point, she's printing out 3D models that are releasing seeds, and she has all this technology that she has connected to a laptop. And the idea is that the squirrel will come up and she will train a squirrel whether to go to a blue or a red circle or triangle. We're not sure which will be most salient yet, to release food. We have a demonstrator squirrel, and then do other squirrels that see this squirrel tend to follow it. And importantly, she's interested in comparing the social network connectivity on social learning and how it varies between the human disturbed site and the less disturbed site. She's interested in whether human disturbance can disrupt animal social learning and culture, if you will. So that's where we're going. That's very much a work in progress, but it should be a lot of fun. Social networks, we have worked out the feeder we're working on. She's working on. I got a great text with some video of it. It's looking really good. Cool. So those are some of the future projects, some of the projects we've been doing with the squirrels. The rest of the talk, I just want to wrap up with some of the big picture ideas that I've been doing from comparative social evolution across mammals more generally. And there are several frameworks that I've been thinking about and trying to explain in various ways. So one is leadership. So I was involved in this working group for several years, thinking about the influence that some individuals have on collective behavior within groups. And in this work, we compared human to non-human mammalian leadership in lots of different domains. I'm not going to talk about this in much detail. I'll tell you a little bit more about some of our recent work. And it just so happens is involved in two separate Phil Tran's special issues. And the first one is on intergroup conflict that came out last year, the end of the year. And my role in that was to look at sex bias in leadership in two major contexts that social mammals deal with a lot. So one is simply group travel, moving from one area to another. So if I were to move as a leader, do I actually recruit followers behind me? Or do I fail to? Only an individual that recruits individuals to follow after is considered a leader. And to what extent does that tend to be a male or a female? The other context is intergroup conflict. So in humans, we might call this warfare. In non-human groups, we think of this as two groups coming in, they each have permanent membership of a society. If those two societies come often at a territorial boundary, meet up, they might have conflict, and that can be lethal at times. And so to what extent do we also see sex bias? And these data are only from non-humans, so I'm calling it sex. And also looking at mammals, we might predict sex differences in these major contexts. In mammals, it's the female that's giving birth, so gestation and lactating, because that's part of the biology that we see. So we might expect that females might be more favored to travel and to safety or to food. We might expect for males to be more mate limited. And we might expect males to be more likely to defend a territory, for example. So those are the bodies of theory that we were drawing from to test whether there's a sex bias for collective movement and or for intergroup conflict. And what we found is when we looked across a phylogeny of different mammals, so I've got some pictures here just to kind of orient you to where we are. The words are fairly small. This is the origin, evolutionary origin, and then the species that are shown here are social mammals that we had good information on both collective behavior in group travel and in intergroup conflict. And what we found is, in fact, sex bias in each of these contexts, but it differed depending on which context we're looking at. So how did it differ? Is a surprise. Here we go. We got, so these are the general patterns that we saw. So this collective movement, this is group travel. We tended to see female bias in that, and that was statistically significant even after controlling for phylogenetic history of these species. So on average, females are initiating and recruiting more followers to safety and to food than were males. However, if we look at intergroup conflict, we see the opposite bias, where there's a significant bias for male-biased intergroup conflict. So what that means is we often only have data that's really robust on participation in these conflicts, but males are participating significantly more than our females in these large conflict events. So we're thinking of this as female guides and male warriors. The idea of male warriors has a strong history in psychology of thinking about the humans actually, and why we might have sex differences in humans and the tendency to go to war across most of evolutionary or historical times, although we have lots of diversity of types of societies in humans today. So this is one piece we'll be looking at. And And we've also found a really interesting part of this is that the degree to which we see this bias tends to be correlated, and I'll tell you how in a minute. But I first want to focus on, so this figure has kind of a lot going on. Collective movements are here on this x-axis. And you see the general trend is that females tend to move and are followed. And we've got 10 species doing that. They also have male bias in intergroup conflict. So this is the average phenomenon. We also see that, in some species, there's a little bit of male bias or no bias in collective movements. But in those species, we also have a male bias in intergroup conflict. So what was really striking and we thought was pretty interesting is that we only saw a female bias in intergroup conflict emerge in those species where they also had a female bias in collective movements. So these are correlated across that phylogeny such that individual species or species, all at species level, species that have female leadership in movements are the only ones that we also saw female biased participation in warfare or conflict, however you want to call it. And I, of course, want to sneak in a little video of spotted hyenas that I mentioned in the start of this talk. They live in these female-dominated societies, and they're really interesting in so many ways. They are unique. Unremarkable is that they will often lead in movement more often than males, because that's the typical pattern. But what is remarkable is they also lead in intergroup conflict. And intergroup conflict for carnivores with big teeth and that weaponry is potentially incredibly costly and can result in injury. So females doing this, that's pretty interesting in my opinion. And what I'm going to show you in this video, it's very messy as these conflicts often are, is that the members on the left-hand side are members of one social group, and then the members on the right are members of a different social group. So here we go, and there might be a little sound.
Okay, here we've got peekaboo hyenas rushing at our guys, and they're rushing back.
And so you might have heard a little bit of a giggle and, laughing hyena. That's actually a signal of submission. There we go. And so, these are really messy events. The females are often at the front. There's a pecking order. Social rank matters. The high-ranking individuals.
Okay, here we've got peekaboo hyenas rushing at our guys, and they're rushing back.
This more often. The males are often in the back. They haven't spent as much time there, and they don't get as much of the piece of the pie when it comes to eating the food that they've cooperatively killed. So they invest less, and it's pretty interesting. In my opinion, I spent a lot of time looking at species that break the norm and try to figure out why that is. So I guess to bring this back to human leadership and why, we might see some gender differences in human societies, I have a paper thinking about, is there an evolutionary explanation for the female leadership paradox? And I would argue that actually, maybe females are leading just as often as males, but maybe the ways that they're leading is quite different. So you might see groups of animals that are moving from one area or collaborative conversations that might not be deemed as leadership as much as maybe going to war or actually, having these really overt interactions. So, that's just kind of an open question, but I'm curious to talk to folks about their thoughts on these things. And we look to the human literature as well as a non-human literature. So just to wrap up, another special issue that just came out in Phil-Trans is on female cooperation. And the role there that I had was to look at within a social group, the extent to which we see males, females, or both sexes cooperating in intra-group coalitions. So I've just shown you this between group conflict. And here we're thinking within a group of baboons, for example, do these two baboons here join forces to direct coalitionary aggression towards another member of the social group? Or do these hyenas join forces to target members of another social group? And do we see coalitions of this type happening in both males and in females equally? There's a lot of literature saying that males form coalitions a lot more than females. So we wanted to test this across mammals in general to see if this is the case. And in fact, what we saw is no evidence of a sex bias with respect to intra-group coalitions. So if we look at the probability of forming a coalition, if we see it emerging in one species in a male, we see it also emerging in females as well. So it's quite equitable with respect to coalitions across the social mammals. And so there's no phylogenetic legacy of a sex bias of forming coalitions within groups. Really fast, quick little work that the final sort of comparative perspective that I've been thinking about most recently is inequality. And, inequality is a huge problem in human societies, but does it actually exist to, similar extents in non-human societies? And if so, what does it look like? So I've been starting to dig at this and look at one form of inequality, which is the transfer of wealth, if you want to call it in human terms, but it's external resources to the individual. So these are commodities that last beyond the lifetime of an individual. It could be stone tools, it could be a territory, it could be food that's stored. And to what extent do we see evidence of wealth or resources being transferred from one generation to another? And we see it, this is beyond mammals. We looked at social insects and fish and all the way up to ourselves. And we see lots of evidence across the tree of life of ways that species are passing on this wealth across different generations. And sometimes this is accumulating to what we're considering legacies of inequality. So for example, if we look on, this is a minimal phylogeny here with some pictures. For hyenas, they might be transferring, for example, these territories that they've invested an incredible amount in, but high-ranking family members are acquiring territories and better positions within them than low-ranking ones. And so this actually gets ramped up across multiple generations, and we see these effects over large timescales. If we go back to squirrels, these are tree squirrels. This is a midden with, this is a big pile of acorns that a squirrel will hoard, and they can choose, these are red squirrels up in Alaska, they can choose whether to give them to one daughter, and if that daughter inherits, we can call her privileged, she's actually set up for life with this stash of food. She reproduces earlier and she has more offspring across her lifespan. Whereas her other daughter that does not receive this midden at the base of the tree, she actually doesn't have as much reproductive success. And so we've been thinking about to the extent to which this inequality is present or absent across the tree of life and trying to look at ecological drivers of it to kind of explain the diversity of when it occurs and when it doesn't. And so what I'm writing, hopefully this weekend, is I'm guest editing a special issue, another one on Philtrans on evolutionary ecology of inequality. And I'm kind of looking at this from the other perspective, like what are the mechanisms that promote equity and equality within these groups and sharing and things like adoption to kind of counteract and level these patterns of inequality that we also see, that are by no means fixed. And we're kind of looking at these different mechanisms that can help to level the playing field. So now that my screen is advancing, I will wrap up very quickly with remind you where we've been. We've talked a lot about ground squirrels, the social decisions that they make, how they have very different personalities and social personalities, also consistent individual differences with respect to predators. I've also then shown you some of these comparative social evolution collective behaviors that we can explain across multiple species. I thank all of the undergrads that are on this team. I remind you, we're looking for more collaborators. My 2 doctoral students are wrapping up. I've got a new postdoc coming on with a new exciting project. Lots of squirrelers interested in doing the work. Thank our funding sources. And I will open it up for questions to the extent that we have time. Thank you for listening. Yes.
Figured out that the social ones are social by themselves. Are they interacting with the same squirrels? The social ones are interacting with the same group of squirrels?
Yeah. So that is somewhat of an open question. My answer is probably yes, because they are faithful to a burrow where they live. So they're similar to central place foragers where, they have that, like birds would do that, come back to a nest. So they're probably interacting with the same individuals. Are some family groups more social than others? Yes. So we definitely have seen that. We could actually tease apart, there are different metrics in the network. So you could look at the degree or how many individuals they interact with. We also do weighted networks. So you can look at the strength or thickness of those connections. And sometimes one of those things explains something and the other doesn't. And so it'd be really interesting to kind of tease that apart. But for the sociality question, individuals tend to be strong on both of those, both the number of partners and also the connections. But it would be interesting, could have loners that are being, they have one partner that they're really faithful to and not spending. Yeah, we certainly see that in humans. So yeah. Thanks. Questions?
Well, we have a couple questions online that I can read.
Oh, perfect. Yes, I have blocked that piece out. So thank you for that. Should I actually get out and look at people?
They can see you.
Okay, yeah.
So
I want to see that.
Right. So if there's could you discuss in greater detail your methods for social network analysis? How do you identify nodes? What might be some of the statistical methods to pursue there?
Yeah. So briefly, if you're interested, I have that recent paper on looking at some of the historical methods that Jean Altman did for her classic behavioral observation techniques, and then updating it with some of the more recent methods of thinking about techniques that we can use. In the squirrels, we know who the individuals are because we live trap them, mark and release. And those are our nodes. So it's whoever we see and we get repeated measures on. And then the tools that we use typically are in R packages. So there's a variety of different R packages that are really helpful, all free to use. And then visualization packages are also in there. So yeah, reach out if you have specific questions. Yeah.
Okay, thanks. Another question is wondering if you have any ideas why a dog would disrupt social networks in your squirrel site, but a coyote maybe wouldn't. And then also wondering why squirrels wouldn't be social given the benefits. What are the benefits for not being social? Why does that persist perhaps?
Yeah. So, at first it was a little bit surprising to see those differences in the social network connectivity in response to coyotes versus dogs. And what we also noticed in that time is if a coyote comes through, the squirrels, they basically freeze and they rear up. If anyone's seen Meerkat Manor and you see, the meerkats looking up and going on sentinel duty, the squirrels do that kind of behavior as well. And they're really quite alert. And so they're pretty steady on their feet when the coyote comes through. And it seems like there's social transmission where on one end of the study site, one squirrel will see it, and then they just go a wave of these vocalizations. And those vocalizations are warning each other that this coyote's there. And it's signaling to the coyote, I see you, don't bother, I know you're there, go somewhere else down the road and find some squirrels that don't see you. The dogs, however, and I should say the coyotes are pretty stealth, they're pretty quiet coming around. The dogs are often quite individualistic, as many dog owners might attest to. Some dogs are off leash, some are on leash. Some dogs are running at the squirrel. So they're literally going to the squirrel, chasing them into the burrow. and standing there. So that is physically disrupting where the squirrels will be. And so what we don't have in the social network is the below ground behavior for this set of information. And in fact, what we're looking at is those above ground networks. So they're probably hiding below ground. And as I showed you some of the earlier work, that hiding below ground takes a while to recover, come above ground, and then forage again. So we think that's probably what's going on. So the intensity of the threat and also how localized it is differs, and the communication systems around it. So actually, we rarely see direct predation. And when we do see it, it's domestic dogs that have killed our squirrels. So yeah, we've seen the other species as well, but it's more likely that dogs are doing it.
Yes, that juveniles were more social than adults and that didn't lose their sexuality as they grow up.
Yeah, that is a really good question. I think it actually ties into the first question we had. So the ways that they're social varies. I've looked at this very explicitly in marmots, and marmots are more closely related to these ground squirrels than the ground squirrels are to tree squirrels. So you might have similar kinds of mechanisms where as the squirrels grow up, they become more selective in who they socialize with. And so they have tighter connections with certain individuals and fewer connections overall compared to the juveniles. The juveniles are often more exploratory. The ways that they connect are also important. So a lot of times adults, they'll just kind of, get up and eat and sit there and, maybe be vigilant and things like that. But the juveniles are doing all these play bouts and these other kinds of things that foster a lot more social activity. So we see that kind of, so the kinds that they do. But interestingly, as they're young, they still have what they're doing there is consistent with what they're doing, that same individual as an adult. So there's some signature of who they will become as they're growing up. Yeah, thank you.
I have one final question.
Yes,
perhaps. So your work bridging into kind of human behavior and society really resonates, but the tradition of applying evolutionary ecology or mammalian behavior to humans has resistance, strong resistance in its academic and professional history. For students or for anyone that wants to take that lens to human behavior, what might you say as they step into kind of the world of scholarship and publishing in that area? And I think it's better now than it has been in the past.
Sure. Well, I think part of the resistance and challenges that there is quite a history of abuse of thinking about these processes and applying them in ways that are harmful to humans. So absolutely recognizing that is very important. In my work, I have been more likely to, what I've tended to do rather than saying, let's look at non-humans and then apply it to humans. I'm actually more interested in how the non-humans are behaving. So what I've tended to do is look at phenomena that are important in humans and see how non-humans are doing it. And then after the fact, okay, is it similar or not? But I'm more interested, I think the female leadership paper got a little bit farther in that domain. I think a really interesting lens is to say, how are people studying humans? Can we study non-humans in similar ways? And do we see actually similarities there? If we are making that connection between pattern seen and non-human animals and going the other way towards applying or thinking about human behavior, it is incredibly important to recognize that there are fundamental differences between humans and non-humans in terms of cultural traditions, ability or free will and choice and all of these things that maybe non-humans have, but we can't interview them, we don't really know. So, even if there is some legacy of it, that doesn't mean it's fixed. There are so much flexibility in human societies to recognize obstacles and these legacies of exclusion and to actually, be proactive to counteract them. So if they do exist to some extent, then, recognizing that that's there is important because it means we need to work even harder to break down those barriers. It's tricky. It's a complicated, yeah. Yep. So Great. Well, thank
once
you.
again.
Thank you so much. Yeah.
Well past time.
Oh, yeah.
Stayed on.
Thank you for staying. Thank you for coming in person. Thank you for coming on Zoom. Thanks for all of your shout outs.
On the chat,
Chat,
I
I
see
see
them.
them.
We'll see you next week, folks. Thanks for tuning in.
Yeah. And reach out.
Meeting data.
Our ideas.
Collaboration.
Cool. Should I stop recording?
Sure.
Okay.